everyone. My name is Akif Eskalen. I am professor of cooperative extension in plant pathology department at UC Davis. Today, I'm going to talk to you about identification of the fungal associates of Mediterranean oak borer. So when you look at the symptomatic plants for the Mediterranean oak borer infested oaks, as you can see here, it starts with this crown thinning and slow dieback starting from the top and going to down. When you see the very advanced infested plants, you often see frass or sawdust accumulated at the bottom of the tree, coming from the galleries that's been infested by the beetle. These are the very common symptoms. When you look a little bit close to those symptoms, you can easily see there are some discoloration, as you can see under the bark on the xylem tissue. You can also see some kind of bleeding, kind of the symptom, which is sometimes could be confused by the Phytophthora root rot, which is very common in California on oak trees. However, when you look inside of the xylem, in this case, as you can see here, you can see the clear discoloration caused by the fungal species. When we first get involved with this project in 2019, at that time, my lab was also working on another invasive shot-hole borer, which is causing the problem still on avocado and other landscape trees and native trees in California. So at first, these kind of symptoms were similar to that ISHP, the symptoms. So that's how we get involved to see what kind of the fungal associate could be causing these tree death. The first thing that we did in this the situation is that identify the symptoms by the beetle itself, and also identify the symptoms caused by the fungal associates. Here's that you can see the base of the one old big oak tree that in the active xylem, you can see the discoloration that possibly caused by the fungal associate of the beetle. So as you may know that most of the ambrosia beetle have their own special organ in their mouth, which is called mycangia, where they usually carry their specific the fungal species. So this is the one on that picture that you see is the female, and this is the male of the beetle. The female is the one that has the special mycangial structure, where she can continue carrying the spore and also making the galleries, infesting those galleries with the fungal species from their mouth, and have them grow in the galleries. And their larvae or offsprings start feeding on these fungal spores or mycelium, and then they move on. So male usually don't have that kind of the structure. They don't have the mycangia, so they just hang around and then just do the mating for the offsprings of the beetle. So female is the one that does all the work. I'm sure my other colleagues in this symposium give you more information about the biology of the beetle. But in this talk, I would like to focus on what are those mycangial spores, how we identify them, and then if they are pathogenic or not. I'm just going to give you a summary of what we have found. First of all, when you are looking for a spore load of the pathogen in the mouth of the beetle, So what we did is that we just remove or chopped their head where the mycangia is located, and then separate it from the rest of the body, and then put them in an Eppendorf tube with the sterile distilled water, and then we just grind them to expose or extract all the fungal species that could be present in the mycangia. So the one on the right that you can see is the very clear picture of those mycangial fungi are growing. So from that, we identified those fungal species based on the not only morphology, but also molecular identification technique using the multi-loci, the sequence of the different regions. So these are the percentage of the recovery of those fungi. So we isolated two kinds of Raffaella, like the fungal species, which are also known as part of the Ophistoma group of the fungi, which is also known as very common the beetle-associated fungi. So as you can see, we isolated them from not only galleries, but also from the mycangia mouth of the beetle. In addition, we also isolated other group of the fungi, Saccharomycopsis, and also we isolated two different Fusarium. One of them seems to be new species of the Fusarium. We haven't named it yet. Another one is the Fusarium solani. We also isolated, as you can see here, from the galleries that we believe secondary invaders or colonizers, which is the Paracilomyces, is the very common the contaminant that we always find infested in the oak tree tissues. So when we look at the literature, Raffaella, which is the Montetia, is the species that we isolated, which is not the new the fungus that we are the only one who identified this fungi. So in the past, in 1998, it was also identified as another beetle, Platypus cylindrus, from France. So another group also identified Raffaella montagiae from the beetle galleries of the Mediterranean oak borer in Germany, which Mediterranean oak borer is native in that region, Mediterranean, the region of the Europe, and also in Portuguese, it was also identified in the cork oak as a Raffaella montagiae. In our case, when we first get involved with the research, we first identify the Raffaella montagiae, Saccharomyces, and other Fusarium and Leptographium that I mentioned. And then these are the ones that we identify from the Mediterranean oak borer infested oaks from Calistoga and Napa counties. So this is the colony morphology of the Raffaella montagiae, which seems to be primary fungal species that you can see their conidia and also conidiophore where they grow. And then most likely the offsprings of the beetle are feeding of these spores and also mycelium of the fungi. So here's another the Fusarium, which we know that it is associated with the Ambrosia beetle. The reason that I'm saying is that so in another the previous work that we have done with the invasive chateau borer is that they are also associated with the special Fusarium species that you can see that their conidia seems to be unique, which we call it dolphin-shaped macroconidia right here. And then microconidia seems to be similar, but this is the one is very distinguishable from the other the Fusarium species that could be present on anywhere else. These are the one that is associated with the Ambrosia. So the next question that was for us to answer is that are they pathogen? So what we did with that is that we inoculated each single one of those fungal isolates on healthy seedlings. Actually, in this case, So we get those seedlings from the white oak, I believe, valley oak, to see if they are pathogenic on those species. And then here are the results. So this is the untreated control. As you can see, there's a small amount of discoloration on the control, which is naturally that part of the plant. But when you look at the other species, right here is the vascular discoloration. Fusarium solani and Fusarium species seems to be the most aggressive, and followed by the Rafaela montegii, as you can see right here, and then followed by the other fungal species. So this is showing us that those primary fungal species that we found associated with the beetle seems to be pathogenic to the plant. I gotta tell you that when you compare the pathogenicity of these fungal associate, they are not that aggressive. Their aggressiveness is about the mild or medium. Therefore, when you have the single infestation of the fungal associate, that's not going to be able to kill the plant moving like the other aggressive pathogen. So they require multiple introduction in the same location by the beetle. Therefore, they could cause the damage by the fungus to kill the plants. Because of that, when the Mediterranean oak borer infests a tree, it takes for a while for plant to die because pathogens are not that aggressive. It's a slow movements. It's always manipulated with the number of the infestation point done by the beetle. So the last part of the work that we also wanted to explore a little bit, if there is any biocontrol agents that could be utilized to manage, prevent, or control these pathogens. In this case, we focus on the bacillus-based the commercial products, and then we did the dual plating with them. As you can see here, that many of those bacillus-based products seems to be inhibiting the growth of the fungi, which is the left side of each plate, and then this is the bacteria. So as you can see, wherever the bacteria grows, they are not allowing the fungal species to get close. So this is the in vitro study. Of course, everything looks good in the laboratory in vitro condition. So the next part of the project should be that the find a way to deliver those bacillus-based products in the plant to provide either the preventative or curative the version of those biocontrol. So we are just the beginning of this side of the project, so we haven't moved to the living plants yet. So with that, I would like to thank you for giving me opportunity. I'm sorry that I'm not going to be able to join this meeting in person. I have other commitments, but here's my email. Those are my lab website. If you have any questions, please feel free to send me email or check those some of those lab works that we have done in our laboratory in our lab website. And then with that, thank you again for giving me opportunity to share this information. And then with that, I look forward to your email if you have any questions. Thank you very much.